In this lecture, we are going to cover transcription, which is when we make an mRNA from a DNA template through base pairing. The skills that you're going to need for the exam is you're going to be able to use a DNA sequence to predict an mRNA sequence. You will also be able to identify the start codon, list all the enzymes needed for transcription, describe the template, also known as the substrate, and the product of transcription, and we're going to compare and contrast the location in the cell that transcription will occur between prokaryotes and eukaryotes. So what I have here on the video already is the central dogma. So this is something that you should absolutely know for the exam, because this is going to be our guide. So this tells us how information moves in the cell. So starting with DNA, anything on this end of the arrow, the left side of an arrow, the arrow is not pointing to, is the template or the substrate. So we can, and then the other side of the arrow, the pointy side, is pointing to the product. Therefore, we can see that in the process of DNA replication that we already covered, we use DNA, because that's at the butt of the arrow, and then this arrow points at DNA again. So we use DNA as a template to make another DNA molecule. And we've already covered this, DNA replication, and those two molecules are identical. DNA is also the template for transcription, this arrow, in which DNA is the template to make an mRNA product. mRNA is then the template for the process of translation to make a protein. So the only part that we're covering in this lecture is transcription, where DNA is the substrate and the mRNA is the product. Okay, so in the process of DNA replication, we know that the enzymes involved were helicase, which was involved in unzipping the two DNA strands so that those bases could be exposed in order to base pair and create two new strands. The enzyme that then created the new DNA molecule, because DNA is a polymer that was a polymerase, and polymerases are always named for the molecule they make. So this is DNA polymerase. That's a review. For transcription, the enzymes used here, again, we're using DNA as a template, and all of the bases are stuck together in the middle. So as you can imagine, we're still going to have to use helicase to unzip the base pairs and expose those bases so that we can copy them. We can use those as the template. The polymerase, however, is different, because in DNA replication we made DNA. In transcription we're making RNA. So the name of the polymer is different. The name of the polymer here will be RNA polymerase, because it's making an RNA molecule. So let's go ahead and work through an example problem. Okay. So this is DNA. We can tell it's DNA because the bases used for DNA, as we did in class, are A, T, C, and G. The bases for RNA are A, U, C, and G. Okay. And in terms of DNA, so we know this is DNA because we've got T's and not U's. That's how we know. In terms of DNA, there's two strands. There is the template strand, which will serve as the template to make mRNA. And then there's the complementary or non-coding strand. Complementary, non-coding. So your book calls it the coding strand, but I think that's confusing, so I probably call it complementary, but sometimes I call it non-coding, because I don't want to confuse you. So that's why there's a vocab difference. It's just because I think this is less confusing. So the template strand is going to make, well, it's not really going to make. This is going to be the template for mRNA. So the template strand is the template for mRNA. The complementary strand is the other one. So if you are given a complementary strand, and it will always be labeled complementary or template, I'm not going to trick you like that. So this is our complementary strand. Then we can determine the template strand based on base pairing. Okay, so the complement's probably going to be 5' to 3', and that makes our template 3' to 5'. Okay, and how do we determine the sequence of our template strand? It's just by base pairing. So A is going to base pair with T. T will base pair with A will base pair with T, C base pairs with G, A base pairs with T base pairs with A, G base pairs with C, T base pairs with A, T base pairs with A, T base pairs with A, T base pairs with A base pairs with T, A base pairs with T, G base pairs with C, T base pairs with A base pairs with T, C base pairs with G. Okay, so now this is the sequence that we can then use to determine our mRNA. I'm going to do that in yellow. Okay, so how do we determine the mRNA? So just like we determined the template strand from the complement, use the template to determine the mRNA. So we're going to completely ignore the complement strand. And the mRNA is going to go 5', antiparallel again, to 3'. Okay, so in mRNA, remember that the A's base pair with U's. So one strategy I do to make sure I get that right, so A base pairs with U, not with T, is I go ahead and I do all of the A's base pairing with U's first so that I don't get confused. And if I have a PhD in the subject and I get confused, perhaps you should do the same trick that I do, huh? Okay, so T is going to base pair with A. Everything else is exactly the same. T base pairs with A. G base pairs with C. C base pairs with G. Okay, and that's how we've taken a complement strand. We can determine the second strand of DNA quite easily. The template, I'm just going to tell you now, is always going to be 3' to 5', so that our mRNA is 5' to 3', because this is the direction that it's read. Okay, and because these are anti-parallel, that means the template is always going to have to be 3' to 5'. I'm not going to flip that around on you. That's too complicated. The mRNA is always going to be 5' to 3'. Okay, so now we're going to talk a little bit about this process. So the RNA polymerase in order to make the mRNA strand, so this makes mRNA strand using template strand of DNA. In order to do this, this binds to a special region of DNA called the promoter. And we're going to see this again later. So you can imagine that the DNA strand is very, 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 very long. And so how does the RNA polymerase know where it needs to make an mRNA. Not all pieces of DNA will make an mRNA, and that's really special for DNA and RNA, is that it has to be told where to start and where to stop. So the RNA polymerase binds to the promoter, which is somewhere up here on the left. Okay. After it binds to the promoter, then it base pairs all the way down until it basically falls off. Then base pairs to form mRNA. Okay, so that's what we did here. And it'll eventually fall off. In the mRNA, there has to be another region that's going to signal where the protein's going to need to start. And that's called the start codon. So the mRNA contains the start codon. And the start codon is the one codon that you absolutely must memorize, because on codon tables it is not always listed that this codon means start. It is assumed that you know it. So that start codon is AUG. You must memorize that. Okay. So how do we figure out where this start codon is in here? There's a whole bunch of letters. Where do we start? Well, just like reading a book, we're going to start at the left, at the 5' end, and we're going to look to the right, and we need these 3 letters in a row. And it doesn't matter, it could be the first 3, it could be the second 3, it could be the next 3. They just have to be immediately next to each other with no other letters in between. So the first codon that we see, that we could potentially see, is AUA. That's not the start codon. The next letter would be UAC. That's not the start codon. So we move to the next one, ACA. That is not the start codon. And we continue doing this until we find it. CAU is not the start codon. But here, look, AUG. This is the start codon. So that's going to be very important for the next step of translation. Okay. So far, we can use a DNA sequence to predict an mRNA sequence. We can identify the start codon. We can list all of the enzymes needed for transcription. And we can describe the template, the substrate, which would be the template strand, and the product, which would be the mRNA, of transcription. So the last point here is that we're going to compare the location that transcription will occur in prokaryotes and eukaryotes. Okay. So I'm going to draw my lovely prokaryote. I'm going to give it a flagella, because why not? And then my lovely eukaryote, which is big. And here we've got the nucleus. Here we've got the cell membrane, which we know is the phospholipid bilayer. And here we've got the cell membrane in our prokaryote. This would also have a cell wall. Let's make that purple. Cell wall. And here we have a cute flagella that would be for movement. Because why not? Okay. So, the DNA in the cell membrane... okay, let me back up for a second. We're going to compare the location that transcription will occur between prokaryotic and eukaryotic cells. So, where will... and I abbreviate transcription TXN. So where will transcription occur? And the answer is where the DNA is located. So why is that the answer? That's the answer because DNA is the template. So anything that uses DNA is going to have to occur where the DNA is, because the DNA doesn't move. So in prokaryotes, the DNA is located in the nucleoid. So that's that dense region of cytoplasm that houses the DNA. In eukaryotes, we know that the DNA that makes you is in the nucleus. So this houses the DNA. So where will transcription occur in prokaryotes? In the nucleoid. That's also part of the cytoplasm. And in eukaryotes, it's going to occur in the nucleus. Okay, so let's quickly compare this to DNA replication. DNA replication occurs in the nucleoid and in the nucleus because DNA is the template for both transcription and replication. So both of these processes, transcription and replication, must occur where the DNA is found. So it really logically makes a lot of sense. Okay, and if you have questions, I hope that you'll bring them to me. And we'll discuss this more during class.